Hey there, pre-AP. Let's talk about some bacteria. So we talked about viruses and about how, in the previous video, about how viruses are not living things because they are considered to be obligate intracellular parasites, meaning that they have to live inside a host cell to be able to reproduce because they bum off our ribosomes to be able to make more RNA or DNA. But bacteria are a completely different deal. And I'm not going to lie to you, I really love bacteria because, it's just so awesome. Hopefully you will find it interesting because there's so many different things about bacteria. Too, I'm just a lover of disease. Not people having diseases. Please don't, but just the study of disease and what they look like and how they infect, human beings and other living things and how you treat them, and it's so cool. So let's get started. I'm excited. Alright, so again, this picture again. So we talked about viruses and how they're teeny weeny. Then here's a bacterium. Love bacteria. Bacteria are a lot smaller than animal cells and plant cells. That's why they can infect those things. So let's get started. Alright, so there are domains of bacterium. There are two. There's domain bacteria and domain archaea. There's eubacteria and archaeobacteria. So let's highlight a few things before I get too ahead of myself. Let's start at the beginning. So prokaryotes, remember, bacteria are prokaryotes. Remember, pro means no. They say no to a lot of things. So let's make a few notes at the top of our paper. They have no nucleus. I know it's been a while since we went over cells. No membrane bound organelles like us. I can't fit that word. And then, what was the other thing they say no to? So no nucleus, no membrane bound organelles. I think those are the two big things. Oh, they're also not fancy like us because they don't have a nucleus. They don't have membrane-bound organelles. But they are really cool, though. Just because they're not fancy like us doesn't mean they're not fascinating. Sometimes the most simple things are the most fascinating. We also need to know some famous bacteria. So let's look at this. Let's highlight some of the most famous bacteria in all the world are the plague that wiped out, more than half of Europe's population in the Middle Ages, 1400s. TB, tuberculosis, used to kill a lot of people. Edgar Allan Poe, if you remember him, his wife and his mother and his aunt, everyone that he loved died of tuberculosis. And then he would always write about how, everybody around him was getting this terrible tuberculosis and dying, and he was wondering why he wasn't, and, later on scientists thought, well, maybe he was resistant to tuberculosis. Maybe his immunity had fought off the bacterium, and he was not susceptible to, getting a terrible case like some of his relatives. Cholera. Cholera is from whenever fecal material gets into the water, and then, people and livestock, use the bathroom upstream, and people downstream drink the water, and then, they got poo in the water. It's not good. Botulism, food poisoning. If you want to put out beside there, food poisoning. You mainly find this in canned goods. So if you ever see a puffy can, don't eat it. If you ever open up canned food and there's fuzzy green or, fuzzy white or just it smells bad, don't eat it. Probably has botulism in it. And then anthrax. Anthraxis bacillus. Anthrax is famous because after 9/11, several buildings in New York City and some random people around the country received these strange letters where they opened up the letter and it had this kind of, powdery white material on the inside, kind of like powdered sugar or flour. And what it was actually anthrax spores. Some bacteria put this protective coat around themselves called a spore, so that whenever the environment's not good for them to grow and reproduce, they'll protect themselves. And whenever, there's enough moisture and the environment's good, that spore will open up and the bacteria will live again. Kind of goes into hibernation. And what happened is they took the letter out of the envelope and kind of shook it to get all the powdery white material off of it, and people who were immunocompromised, people who were very sick or elderly or what have you, they would breathe in those spores, and then some of them, I think there was one elderly man or woman in Florida that actually died from it. And there were buildings in New York City that received these letters, and they had to have hazmat suits called in. It was crazy. But this was right after 9/11, and, we were all on we were a little paranoid of everything that happened. Crazy stuff happened. So, but please, let's highlight, most bacteria are harmless, though. Some awesome things that bacteria do for us. If you want to write this down, some examples, things like yogurt and cheese. We would not have cheese without bacteria, people. We wouldn't. We wouldn't have yogurt. It would be bad. We also need to highlight that they are essential for life to exist, because a lot of bacteria, they help us in nutrient cycling, like nitrogen, and they're also decomposers. They break down the dead organic material, which is good. So there are 2 domains. Please, let's highlight bacteria. These are the common ones. They live in everyday places. And then archaeobacteria. These are extreme. These are called extremophils. They live in extreme places. So there are 3 bacteria shapes or structures. Please highlight that if you're a prokaryote, you're unicellular. If you want to put out beside there, that means 1 cell. And there are 3 basic shapes, so we're going to draw them. First one is called a cocci. These are round. This is an example of a cocci bacteria. It's a staph infection, or Staphylococcus aureus. What bacteria, whenever they are named for usually the shape that they're in. So Staphylococcus aureus, if you want to put that, is actually, staph means cluster, and then coccus means round. So it looks like a cluster of grapes, if you want to put that out there. Looks like a cluster of grapes. Staphylococcus aureus. I probably spelled that wrong, but it's okay. For learning purposes, if I find the really good spelling. Sorry, guys. So, round-shaped bacteria. Example, Staphylococcus aureus. The next kind is bacilli. Bacilli are rod-shaped, and a very good one is actually anthrax. Anthraxis bacillus. And it's just little rod shapes. Let's draw some rod shapes. They're kind of round. Some of them come in chains. Some of them come in pairs. So just draw you some rod shapes. Anthraxis bacillus is your example. There's actually called, tapped it too many times, helical. Sometimes you would call this spirillus. So have you ever, let's put that out there, AKA spirillus. Sometimes it's called spirillus. They're helical. They actually look like this, so let's draw them. I don't know many examples for helical bacteria or spirillus. These are kind of not the most common. So let's also highlight after our shapes. Most prokaryotes have a cell wall, and you can also identify bacteria by how thick their cell wall is. So let's go why we have a cell wall, just kind of like in plants. Let's highlight primary for protection. because you're going to protect that cell membrane. And it also prevents the prokaryote from bursting from an aquatic environment in case that bacteria, lives in water. So that cell wall is mainly made of proteins and sugar. And we need to highlight this special word, peptoglycan. Peptoglycan is a sugar. So, depending on how thick your peptoglycan layer is in your cell wall, tells us some things about you if you're a bacteria. So let's highlight gram staining. Gram staining allows us to see what type of bacteria, if in a lab, if you take a sample from a patient, you think, oh, they probably have a bacterial infection. I want to find out what bacteria they have. You're going to put a series of dyes on that bacteria sample, and you're going to look at it under a microscope. So this is for fast, easy identification. So let's highlight. Scientists perform gram staining for easy, fast identification of most bacteria. If you are gram positive, this guy right here, gram positive, you actually stain blue, or put this out beside there, or purple. It'll either be really dark blue or pretty much purple colored. And why do you stain blue? Because you have a thick cell wall of peptoglycan. What it does is the first stain that you would put on that bacteria is called crystal violet. And what that thick cell wall does is it soaks up that purple crystal violet, and then it'll just stay there. It's like a sponge. It will not absorb the next few dyes that you'll put on it. Now, let's say I'm not gram positive. I'm gram negative. That means that I have a thin peptoglycan layer. And I will stain red and then put out beside there, or pink. What happens is, why this turns pink or red is because that layer of peptoglycan is so thin that if you put the crystal violet on it first, it won't really soak it up like that thick wall does. And after you wash it off, you'll put this other stain on it that's red. I can't remember the name of it. And what it will do is, it's a little bit smaller molecule, and it will actually absorb the red. And depending on how much it absorbs, it'll be really red, or it'll be kind of pinky red. And why we want to know this is because depending on how thick your cell wall is will tell us what type of medicine will best treat that bacteria. The ones that are most dangerous to human beings are gram negative. So let's highlight that, please. Gram negative are more dangerous to humans because they can be resistant to antibiotics. Why they can become resistant? They can actually change their DNA. They can mutate a lot easier than those gram positive. We need to highlight that some bacteria produce a capsule that covers their cell wall. It is a sticky substance for adherence to surfaces. And we need to highlight what's kind of most people identify it with is it's what actually makes people sick, not the bacteria. An example of that would be, please put out beside there, an example of this is E. coli. E. coli, whenever it gets into your stomach, like food poisoning, if you want to put out beside there, is it's not the bacteria itself that makes you sick. It's the toxins let off from the E. coli capsule. So, just some interesting facts. So, bacteria movement, we need to highlight that some have flagella, a whip-like tail, like in this picture. Some have cilia, kind of looks like little bitty, kind of fuzzies all around it, like a sucker dropped in a carpet. We also need to highlight in that next letter that some prokaryotes move by sliming. What they do is they kind of spit out a layer of slime, and then they kind of and slide all over it. Alright, taxis. What this means is it's the response to a stimulus, or, this is pretty much used with any organism. If it's positive, it indicates that it moves forward. If it's negative, it indicates that something moves away. So usually what we do, the stimulus that is used, go and highlight that it's usually light, gravity. Sometimes we put a current, like a wavelength, out beside it and see what it does. And then chemicals, how it reacts to chemicals. Genome, we need to highlight that they are a singular, circular strand located in the nucleoid region. Remember, they don't have a nucleus. They just have free-floating DNA, a nucleoid, like in this picture. And then we need to highlight the very last statement in that. Prokaryotes also have plasmids for exchanging. This is how good old bacteria can become resistant to antibiotics. So remember, we kind of talked about plasmids with DNA cloning. Remember, we cut out with a restriction enzyme, make 2 sticky ends, stick in the plasmid, that bacterial DNA, move on. How bacteria reproduce, they don't go through mitosis or meiosis. They don't. So let's highlight. Bacterial reproduction is accomplished by the process of binary fission, which is to make clones. It's an exact copy of the parent. It is specifically identical to the parent. No variation. So please highlight that this is asexual reproduction. That means, put out beside there, no gametes. Doesn't require gametes. All that bacteria does is it grows so big, divides its cell membrane down in half, and then 2 new daughter cells. So means of nutrients, why we like bacteria, it helps us cycle nutrients. So let's talk about the ones that help. So saprobes, they eat dead material. They're decomposers, very important for us. Of course, there's parasites. Those are the ones that harm us, make us sick. Nitrogen fixation. This helps in the nitrogen cycle. Just put that out beside there at the end. They help take ammonia and produce nitrite, which is, helps us cycle nutrients through the soil, helps us grow things. And nitrification, that's whenever you take the ammonia and then you produce nitrite. And then photosynthesis, they use the sunlight so that they can, make some sugar if they're photosynthetic. Chemiosynthesis, they take hydrogen sulfide and they make energy. Heterotrophs, they just feed on another organism. So here they are again in a nice pretty table if you want to look over that. So let's move on to part 2 real quick. It's really short. So oxygen demands, before we get to the methanogens, oxygen demands, please highlight that. If you are a bacteria, you can live in one of 3 environments. You can be an obligate. Think about what obligate means. Remember, we talked about that with viruses. Obligate means you need it. Aerobe. Aerobe means air or oxygen. So please highlight, they must intake oxygen to utilize their primary source of energy. They have to have oxygen to live. Maybe you're a facultative anaerobe. Remember, a or an, what does that mean? These organisms can have both. They can function with or without oxygen. They kind of, have the best of both worlds, as Hannah Montana said. Ooh. Obligate anaerobes, these are things like botulism. They must be without oxygen. That's why botulism can live in canned food, because there's no air in there. So, let's also highlight terms used in archaea or extremophils, where let's talk about some extreme bacteria. Extreme. So one of them is a methanogen, and why we call them methanogens is because they produce methane gas, and they're mainly associated with ruminants. These are animals with a rumen, or it's kind of part of the stomach. Cows have this, where because they eat grass and they have to break down that cellulose in the cell walls. And let me see. You will also, if you ever, some people I've heard before in my classes go, well, Miss Minor, like, we talk about, methane, whenever someone passes gas. Yes, that is methane gas whenever you break wind. Halophils, these guys love salt, so please highlight, these are salt lovers. They live in places, examples, they live in places like the Dead Sea. No organisms will live in the Dead Sea. No plant, fish, animal, nothing. But this bacteria will. And then thermophils, these are heat lovers. They love to live in hot springs. They also love to live in underwater sea vents in the ocean.